So this lecture is going to focus on chemical polypeptide synthesis. Much of it will be on the board. So the problems are that in chemical polypeptide synthesis, there are 20 different building blocks. All building blocks are connected by amide bonds, so you can't simply mix all the needed amino acids and allow them to react randomly. This next part, as I've told you, is on the board, so you'll have those notes. For amino acids that have reactive side chains, there are protecting groups that can be used, and here's a table of side chain protecting groups and the reagents used to protect the groups. So here you have tert-butyl, benzyl, trityl, methyl ethyl, and this pentamethyl chroman 6-sulfonyl group, and here are the groups that they protect on the subsequent row. And so this is how you would introduce these protecting groups, and then these are the cleavage reactions. And so in an exam or problem set, you should be able to use these protecting groups in order to build your peptides. Again, making sure you use appropriate stereochemistry and noting when to add these protecting groups and then how to cleave them off. So in solution phase peptide synthesis, there are two ways of achieving selective deep protection. One is to choose protecting groups that are deep protected with different reagents. This is known as orthogonal protection. Or the other way is to choose protecting groups that are deep protected with the same type of reagent but under different conditions. Shown here is the synthesis of serine, alanine, valine peptide, tripeptide, starting off with valine, adding this Bach alanine, and then creating this dipeptide. And then the acid is used to deprotect the Bach group and alanine. And then you're adding another serine, and look, as from the previous slide, the serine is protected with this tertiary butyl group, and so in future you should know how to add the tertiary butyl group. And then in the presence of diisopropylethylamine, you make the Bach serine alanine valine, and then you're using methanol and sodium hydroxide as followed by the acid. So the methanol will probably deprotect, and then the acid to deprotect also the Bach protecting group. Bruce Merrifield, who won the Nobel Prize in 1984, developed solid phase peptide synthesis, or SPPS. The process is you have an amino-protected amino acid coupled to a solid phase resin, shown here. Protecting group is removed, and then another amino-protected amino acid is added. Repeat steps 2, 3, ending with step 2. Then cleave the polypeptide from the solid resin. And so the advantage is that you can make this can be now fully automated, and the byproducts are easily rinsed. So here's Merrifield synthesis, in which the resin is a polymer, styrene divinyl benzene resin. Here you have your resin, your amino acid. You couple the amino acid onto the resin, and then you start deprotecting, adding another amino acid by activating the amine, and then coupling to generate the polypeptide still attached to the resin, HF to deprotect and cleave off the resin. FMOC synthesis is actually the most commonly used type of SPPS, in which you start off with a resin and an FMOC amino acid. Once you have that first amino acid attached, you deprotect, right, remove the FMOC, and then you have the amine. You're going to activate, add another FMOC amino acid, and then you have a coupling happening, and you can go through this cycle of acylation, FMOC amino acid, and then wash And then when you're ready with your polypeptide, treat it with, this is trifluoroacetic acid, and this is the mechanism by which you can remove it off the resin upon treating with the acid. So, the nice part about this is that you can acylate in a recirculatory move as well as wash to remove excess agents. In linear polypeptide synthesis, you add one to make the dipeptide, add another one to make the tripeptide, add another one, tetra, add another one to make the penta, and then add another one to make 6, the hepta. There's another way of which you can make polypeptides, and this is through convergent polypeptide synthesis, in which you start coupling 2 of each. So you have 1, 2, 3, 4, 5, 6, and then you couple one set together, and then you couple the last set. And then the advantage of using convergent polypeptide synthesis is that there are fewer steps, and you can result in large yields because each step will lessen the yields. So if we look at a convergent polypeptide synthesis example using solid phase peptide synthesis, here's one which makes segment 2, another one which makes segment 1. You condense, you form the dipeptide, and then you do solid phase peptide synthesis to make segment 3, and then you couple so that you generate the polypeptide. HPLC, or high performance liquid chromatography, is used as an analytical technique for solid phase peptide or peptide synthesis. Separation techniques involves mass transfer between stationary and mobile phase. Your liquid mobile phase is used to separate the components of mixtures, and analytes are dissolved in the solvent and then forced to flow through a chromatograph column under high pressure. So in HPLC, physical absorption usually takes place through surface attraction and repulsion forces. Here are the forces involved. You have hydrophobic or dispersive interactions, London-type dispersion forces, interactions between induced or permanent dipoles of molecules, and the electric field of the absorbent surface, charge transfer between the adsorbed molecule and the adsorbent. There are different types of HPLC. Each HPLC mode possesses its specific type of interaction. So for reverse phase chromatography, that mainly uses a non-specific hydrophobic or dispersive interactions. And this is actually commonly used for solid phase peptide synthesis and purification. Normal phase mode mainly utilizes polar interactions, dipole-dipole, and dipole-induced dipole. And then you have ion exchange chromatography, which employs an ionic interactions of ionizable analytes with ion exchange adsorption centers on the absorbent surface. In reverse phase HPLC, which is used in commonly used for peptide purification, your polypeptide enters in the mobile phase, it adsorbs on the reverse phase surface, and then desorbs from the reverse phase surface when the organic modifier concentration reaches a critical value. So in general, for polypeptides, you use water acetonitrile as the mobile phase with TFA to protonate so that it becomes hydrophobic. There's also cyclic or constrained peptides. And in cyclic or constrained peptides, you make a linear peptide and then you cyclize it through different types of bonds. Here's a disulfide bond. Here is also a disulfide bond within a polypeptide chain. And then you can do head-to-tail cyclization. So the reason why cyclic peptides are made is because you're trying to mimic cyclic or constrained peptide fragments that exist in natural proteins. Here's an example of an antibody, and you can see all of these loops that are responsible for recognizing antigens and binding of antigens. And so being able to mimic these loops is important. There are different synthetic strategies for cyclic or constrained peptides. So here's a linear peptide, R1 to R5, and you can have, for example, the amine and the side chain of R3 interacting with one another. you have through the side chain of R2 making the cyclic. You can have just a backbone being cyclic. You can have the R2 and R4 interacting with one another and forming cyclic polypeptide. And then you can have R5, or this is R6, sorry, bridging and cyclizing in this mode. There are four main strategies for cyclic or constrained peptides. You can have a solid support attached to a C-terminus through a safety catch linker. You can have a solid support attached at the C-terminus through activated linker, and solid support attached to peptide through a side chain, as well as solid support attached to the peptide through an amide bond. So here is an example of synthesis of cyclic peptides. Here you have your resin, your first amino acid added on, and then your second amino acid added on, and then your growing polypeptide chain, right, that through solid phase peptide synthesis. And then through palladium, you have a cyclization occurring in this, where you have your amine, and then this gets removed, and then through HF, cleave it off the resin, showing the cyclic peptide being generated. This mode of synthesis is called backbone linker approach. This is another example, in this, which uses an activated linker. Again, here's your resin. Here's your starting moiety, in which you do solid phase peptide synthesis and grow the polypeptide. And then from here you have intramolecular cyclization, kicking this off, forming this amide internal bond. Another method, and this is attaching through the side chain, is shown here. Again, here's your resin, you're growing your polypeptide chain. Here's your polypeptide, and now you're going to do intramolecular cyclization in the presence of this group, and then once it cyclizes, you have now a cyclic peptide in this way. So this uses a ring contraction strategy for synthesis of difficult cyclic peptides. Here you have your peptide sequence. Now you're using BOP, and so now what's happening is you're closing this in and stabilizing this so that you can allow for intramolecular cyclization, forming the cyclic peptide, and then through light removing this to generate the ring. So it's literally you're using a larger ring to contract and stabilize the other ring. The other important thing is that alpha helices and beta sheets are used for molecular recognition, and here's the examples. So here's a basic helix loop helix protein, USF, binding to the major grooves of DNA, where you can see the helix actually interacting. Here's a helix turn helix protein, MAR-A, bound to DNA, and you can see these helices are important in interacting with DNA. And then you have the TATA binding protein, which we discussed in earlier lectures. Now in this case, you have this beta sheet that recognizes the TBP, that recognizes the TATA binding site. So if we think about the example of the helix turn helix motif, you have all of this protein, but the real important part of the protein that's doing the molecular recognition is shown here. So if you remove this alpha helix from its context, it then becomes unstructured because it doesn't have all of this to stabilize the helix. And as a result, it doesn't bind as well. And so even though this is where all the interactions for the DNA are, just taking that isolated alpha helix will not allow it just to bind, because it will have to use energy in trying to make the helix to allow it to bind. So because it's minimally structured and functions poorly, because it has to pay this entropic cost. So cyclization or stabilization becomes an important feature for making these short peptide helices. So you can use non-natural amino acids, pi stacking. Here's metal molecular recognition, disulfide bonds, lactam bridges, carbon-carbon bonds, and then helix initiator examples. There are also beta sheet and beta hairpin stabilizers. By incorporating D or L proline, you can favor formation of antiparallel turns. You can put D proline for connecting C and N with C with diamine and N with urea termini to again stabilize the hairpin for beta sheet, beta hairpin assemblies. You can use this organic moiety and then connect peptides to stabilize and form beta hairpins. And shown here is an example, as well as you can make a backbone cyclized version. So far, we've discussed peptides and strategies for building peptides to mimic biology. But also, peptide can self-assemble and form interesting structures and unique nanostructures and surface materials that can either attack cell surfaces and make holes through it, punch holes through it, or form gels, as shown here as an example. So here's cyclic peptides that have been built by the Gadiri group, which can self-assemble into these tubes, these nanotubes, which can then be used as antimicrobial agents. Here's another peptide in the presence of light can assemble into these beta sheets or beta hairpins, which then assemble into these hydrogels, and then these can be used as scaffolds for tissue engineering. And so protein engineering, in terms of using peptides, can be used not only to mimic and try to reproduce biology, but also to make new types of materials that can impact biology, or that can be used to engineer biology.